Imagine your best friend, your dog, part of the family just isn't themselves. Maybe they're dealing with these persistent, kind of mysterious health problems.
Yeah, things like vomiting again and again or ongoing diarrhea, maybe losing weight for no clear reason.
It's truly awful to watch, and for any dog owner, it's just deeply worrying.
It absolutely is. And these signs, these classic frustrating signs, often point towards something called canine chronic inflammatory enteropathy, CIE for short.
She,
Yeah,
right.
it's a common chronic gut issue in dogs. It can hit the small intestine, the large intestine, sometimes even the stomach. It's a real puzzle.
And that puzzle is exactly why we're doing this deep dive today. Our mission is really to cut through some of that complexity.
Mhm.
We want to help you understand the latest research, the really cutting edge stuff about how your dog's own immune system, both the fast acting innate side and the more specific adaptive side, might be tangled up in this condition.
And we'll also look into the search for biomarkers.
Right, those crucial clues that could maybe help with diagnosis or predicting outcomes or even finding totally new treatments down the line.
It's a journey into veterinary science frontiers, really. Some surprising things are coming out.
Okay, so let's start by defining CIE a bit more clearly. What exactly are we talking about?
Right. So CIE refers to gut problems, gastrointestinal issues that have been going on for 3 weeks or more. That's the chronic part.
3 weeks, okay.
And crucially, before you land on a CIE diagnosis, you have to have ruled out other possibilities.
Like what kind of things?
Oh, infections, parasites, even cancer or problems outside the gut that look like gut problems. It's what we call a diagnosis of exclusion. You rule everything else out first.
Got it. So when people hear IBD or inflammatory bowel disease for dogs, they probably think of human conditions, right? Like Crohn's or ulcerative colitis. Is canine CIE basically the same thing?
That's a really common question, and actually, no, it's where things get quite distinct. While the names sound similar, canine CIE is different.
How so?
Well, a key difference is how we classify it in dogs. We do it clinically, based on how the dog responds to treatment.
Oh, okay. Based on response.
Exactly. So first you have food responsive enteropathy, or FRE. These dogs, thankfully, get better just by changing their diet.
That sounds like the best case scenario.
It often is. Then you have antibiotic responsive enteropathy, RE, where, as the name suggests, antibiotics help.
Now, using antibiotics for gut issues, that sounds like it could have downsides too.
You're right to flag that. It's really important. Just handing out antibiotics empirically, sort of, just in case for R, can actually damage the dog's gut microbiome long term.
So needs careful thought.
Definitely. Ideally, antibiotics that are only used after a really thorough checkup, maybe if other treatments haven't worked or if there's solid proof of specific problem bacteria causing trouble. You don't want to disrupt that gut balance unnecessarily.
Makes total sense. Okay, so we have FRE and ARE. What comes next?
Next up is immunosuppressant responsive enteropathy. That's IRE or sometimes SRE. These cases need medication to actually suppress the immune system's overreaction to control the inflammation.
Okay, so that's a step up in treatment.
It is. And then, unfortunately, there's non-responsive enteropathy, or NRE. These are the toughest cases because they just don't respond well or at all to the standard treatments we have.
Wow, that must be incredibly challenging for everyone involved. So classification is based on treatment response. Are there other ways vets look at it?
Yes, there's also histopathology. That means looking at tissue samples under a microscope.
Biopsies, right.
Exactly. That tells us which part of the gut is inflamed stomach, small intestine, large intestine, and what type of inflammatory cells are causing the trouble. Maybe lymphoplasmacytic cells or eosinophils.
Yeah.
That gives us clues too.
So different cell types might be involved.
Right. But here's another really important difference from human IBD. There's no direct equivalent to Crohn's disease in dogs. And, critically, the treatment approach differs. Surgery, which is common in human IBD to remove damaged gut sections, is not typically indicated for canine CIE.
That's a huge difference. So we know how it's classified: response, cells, location. But the big question, the reason for this deep dive, is why? Why does CIE happen? You said the exact cause is still a bit of a mystery.
That's the core issue, yes. Despite all the progress, the pathogenesis, the actual mechanism of how it develops, isn't fully understood. It's clearly multifactorial.
Meaning lots of things play a role.
Exactly. Genetics definitely play a part, the dog's environment, the gut microbiota, that massive community of bacteria living in the gut, and crucially, a dysregulated immune response. The immune system just isn't behaving correctly.
And that's why zeroing in on the immune system is so important here.
Precisely. It's probably one of the biggest pieces of this whole puzzle.
Okay, let's start breaking down that immune response then. First up, the innate immune system. This is like the body's first responders, right? Quick, nonspecific defense.
That's a great way to put it. The immediate emergency crew, its role in CIE is, well, complicated, and we're still figuring out all the details.
And you mentioned the gut microbiota being involved. How does that connect to this first line of defense?
It's a huge connection. We consistently find something called dysbiosis in dogs with CIE.
Dysbiosis, meaning an imbalance in the gut bacteria.
Exactly. A significant imbalance. It's strongly linked to CIE. There's even a tool vets can use now, a dysbiosis index, to help measure these changes.
Interesting. What kind of imbalance are we talking about? What changes?
Well, studies show that dogs with CIE often have fewer of the beneficial bacteria, things like Clostridia and Faecalibacterium species. These guys are important because they produce stuff like short-chain fatty acids, particularly butyrate, which actually helps reduce inflammation.
So the good guys decrease.
Right. And at the same time, you often see an increase in potentially problematic bacteria, like proteobacteria. It's like the helpful residents are moving out and less desirable ones are taking over.
Okay, so the bacteria balance is off. What about the gut lining itself? You hear about leaky gut. Is that relevant here? The mucosal barrier?
Absolutely relevant. Think of the gut lining as a fortress wall, like you said. It's got layers of mucus, physical barriers. Its job is to keep the gut contents, food, bacteria, toxins, safely inside the gut lumen and away from the body's tissues.
The critical defense line.
Totally critical. But in dogs with CIE, this barrier can break down. It becomes leaky, compromised.
Like cracks in the fortress wall.
Exactly. And when that happens, you get increased inflammation right away. Plus, the immune system loses what we call immunological tolerance.
Meaning it starts attacking things it shouldn't.
Precisely. It starts overreacting to normal, harmless gut bacteria that it would usually ignore. These bacteria, or parts of them, can then seep through the leaky barrier, getting into the tissue and triggering a much bigger inflammatory response.
Are there specific molecules involved in this barrier breakdown or repair process?
Yes, there are. For example, trefoil factors, or
Yeah.
TFFs. These are proteins made by special cells in the gut lining called goblet cells, and they're important for healing and repairing that barrier.
Okay, TFS for repair.
Right. But in CIE dogs, the pattern is weird. TFF1 levels might be up in the duodenum, the first part of the small intestine, but TFF3 levels are often down in the colon. So the repair process seems disrupted, maybe not working properly everywhere.
That's complex. Any other key molecules?
Another interesting one is P-glycoprotein, or PGP. Think of it as a tiny pump on the surface of cells that pushes unwanted substances out.
Like a bouncer for the cell?
Kind of, yeah.
Yeah.
And its expression, how much of it there is, can be abnormal in some dogs with a specific type of CIE called lymphoplasmacytic enteritis.
Okay.
But here's something really significant.
Mm-hmm.
Some research suggests that if PGP levels increase after treatment with certain drugs, like prednisolone.
Common steroid, right.
Exactly. If PGP goes up after that treatment, it might actually predict that the treatment is going to work well for that dog.
Wow, so PGP could be a predictive biomarker for treatment response.
Potentially, yes. It's a really exciting lead towards more personalized medicine for these dogs. Instead of just trying things, we might be able to predict success.
That would be huge. Okay, so we have the bacteria, the leaky barrier. What about the actual immune cells rushing to the scene? The innate immune cells?
Well, neutrophils are a big one. They're often the very first white blood cells to arrive at inflammation. We see more of them in the duodenum of CIE dogs, and higher numbers often correlate with how severe the disease is.
Are there ways to measure this neutrophil activity?
There are potential biomarkers being looked at, things like serum PNCA, it's an antibody test, and also the neutrophil to lymphocyte ratio, or NLR.
NLR.
Yeah, just the ratio of neutrophils to another type of white blood cell, lymphocytes, in a blood sample. It's simple, but it seems quite useful. It might help differentiate between the food-responsive type, FRE, and the immunosuppressant-responsive type, IRE.
Which could help guide treatment choices earlier on.
Exactly. Saves time, potentially avoids unnecessary treatments.
What about other innate cells, like macrophages? They're kind of the cleanup crew, aren't they?
They are, but they can also drive inflammation. In CIE, we often see more macrophages in the gut tissue. And importantly, there seems to be an imbalance. More of the pro-inflammatory type, called M1 macrophages, are present.
The ones that turn up the heat.
Right. And this is often linked to increased activation of a pathway inside the cells called NFB. Think of NF key as a master switch that turns on a whole bunch of inflammatory genes.
So the heat switch is stuck on?
In a way, yes.
Yeah.
And speaking of markers, here's a really promising one. Calprotectin.
Calprotectin, I've heard that mentioned.
It's a protein that gets released mainly by neutrophils and macrophages when they're activated, and it's found to be overexpressed in the gut lining of dogs with CIE.
So more inflammation means more calprotectin.
Generally, yes. And the exciting part is that we can measure it in feces fecal calprotectin. It's considered probably one of the most promising non-invasive biomarkers right now for figuring out how severe the gut inflammation is in dogs.
Non-invasive is key for owners and dogs. That sounds incredibly useful. What are the hurdles before that becomes a standard test everywhere?
Well, the main challenges are standardizing how samples are collected and tested across different labs and clinics, and establishing really clear cutoff values. What level means mild, moderate, or severe disease? It needs a bit more validation for widespread use, but the research is definitely heading that way.
Good to know. Okay, any other innate cells we should touch on? Eosinophils, mast cells.
Yes, eosinophils are often increased too, especially in certain types of CIE, like eosinophilic enteritis. We even find degranulated ones in the gut tissue.
Degranulated meaning they've released their inflammatory contents.
Exactly, they've dumped their cargo. And canine eosinophilic gastroenteritis actually looks quite similar clinically and under the microscope to the human version.
And are there markers for these cells too?
There are potential non-invasive ones being explored. For eosinophils, something called serum 3-bromotoracine, or 3-BRY. For mast cells, N-methylistamine, or NMH. Measuring these could give clues about eosinophil and mast cell activity without needing that invasive biopsy.
More non-invasive clues. Excellent. We're also hearing about things called S100 proteins or calgranulins. What's their story?
Right. S100A12 is one specific type of calgranulin protein. It's another one that seems clinically useful as a marker for GI inflammation. Higher levels in the feces are strongly linked to disease activity and how severe it is.
So it's similar to calprotectin in a way?
Similar concept, yes, released during inflammation. And this S100A12 ties into another system involving something called the receptor for advanced glycation end products, or RAGE.
RAGE, okay.
It's a receptor on cells that S100A12 can bind to, triggering more inflammation. In CIE dogs, the pattern is interesting. They seem to have less of a soluble, free-floating form of RAGE in their blood, S-RAGE, but more RAGE expressed on the cells lining their gut.
So less of the decoy receptor floating around and more of the trigger receptor on the gut cells.
That seems to be the implication, yes. It suggests this whole RAGE system is dysregulated, and maybe targeting it could be a future treatment strategy to help calm things down.
Fascinating potential there. Okay, one last piece for the innate system. Pattern recognition receptors, PRRs. What are these?
PRRs are like the immune system's molecular sensors, or sentinels. They sit on immune cells and gut lining cells, and their job is to recognize specific patterns found on microbes, like bacteria. Think toll-like receptors, TLRs, or NOD-like receptors, NLRs.
So they're meant to spot bacteria.
Yes, and crucially, they usually help the immune system distinguish between harmless commensal bacteria, the normal residents, and dangerous pathogens, helping to maintain that tolerance we talked about.
But in CIE, something goes wrong.
It seems so. In CIE, these PRRs appear to misinterpret signals. Instead of recognizing normal gut bacteria as harmless, they trigger an inflammatory alarm bell.
Leading to that inappropriate inflammation.
Exactly. And what's really interesting here is the genetic link. We're finding that variations, polymorphisms in the genes for some of these receptors like TLR4, TLR5, and NOD2 are associated with a higher risk of CIE in certain breeds.
Like German Shepherds?
German Shepherds are a key example, yes. There's a specific variation, a haplotype, in the TLR5 gene that's been linked to more severe inflammation in that breed. It suggests some dogs are just genetically wired to overreact.
Wow, okay. So the innate system is clearly incredibly active and complex in CIE.
Yeah.
If you had to boil down the absolute key message from this part for a dog owner listening, what would it be?
I'd say the main takeaway is that your dog's first line of defense, the innate immune system, can unfortunately get stuck in overdrive in CIE. It overreacts, especially to the gut bacteria. But the good news is that markers like fecal calprotectin are giving us exciting, non-invasive ways to actually measure this internal battle, hopefully leading to faster, more accurate diagnosis and monitoring.
That makes sense. A system designed to protect ends up contributing to the problem, but we're getting better at seeing it happen.
Precisely.
Okay, that sets the stage perfectly for the next layer. The adaptive immune system. This is the more specialized force, right? The one with memory.
Exactly. The specific fighters, the ones that learn and remember threats.
Yeah.
Slower to respond initially, but much more targeted.
And who are the main players here?
Well, the key commanders are the T helper cells, also known as CD4 plus T cells. You can think of them as directing different types of immune responses. There are several main teams.
Teams? Like what?
You've got Th1 cells, Th2 cells, Th17 cells, and then the regulatory T cells, or TREGs.
Okay, what do these different teams do, broadly speaking?
So, very basically, Th1 cells are crucial for dealing with intracellular pathogens, like viruses or certain bacteria. Th2 cells are classically involved in fighting parasites, but also heavily involved in allergic reactions. Th17 cells are potent drivers of inflammation, particularly important for defense against some bacteria and fungi and mucosal surfaces. And then TREG cells. They're the peacekeepers.
The peacekeeper.
Yeah, their main job is to suppress immune responses, to prevent the system from overreacting, maintain tolerance to self-antigens, and keep everything in balance. They're incredibly important.
Now, I remember you saying that in human IBD, there are often distinct profiles, like Crohn's might be more Th1, Th17 driven, and ulcerative colitis more Th2. Is it that clear in dogs with CIE?
That's where it gets tricky again. Unlike in humans, we haven't really been able to pin down a single, clear, predominant T helper profile for canine CIE.
Why is that?
It largely comes down to the heterogeneity of the dogs studied, the different clinical presentations, FRE, IER, the different breeds, diets, environments. It makes it really hard to find one consistent immune signature across all CIE cases.
So it's more varied in dogs, or at least appears that way in studies so far.
It seems so. What we have seen, though, are bits and pieces. Some studies find a mix, sort of a balance of both pro-inflammatory and anti-inflammatory signals, or cytokines.
Cytokines being the chemical messengers.
Exactly. Other studies do show clear increases in pro-inflammatory cytokines like interleukin-2, IL-2, and tumor necrosis factor alpha, TNFA, which definitely push inflammation forward.
Any other specific findings?
One intriguing finding is in dogs with the food responsive type, FRE. Some research shows decreased expression of something called IL-33 mRNA in their gut tissue.
IL-33, what does that suggest?
Well, IL-33 is often associated with Th2 responses. So seeing it decreased in FRE dogs might hint that their Th2 pathway is behaving differently compared to what's seen in some human IBD, like ulcerative colitis. It's another piece suggesting canine CIE isn't just a direct copy of the human condition.
Interesting. What about the pro-inflammatory Th17 cells?
We do see evidence for their involvement. Increased levels of IL-17A, which is the signature cytokine for Th17 cells, have been found in some cases. And perhaps relatedly, some studies report low numbers of those crucial TREG cells, the peacekeepers.
So more inflammation drivers, Th17, and fewer peacekeepers, TREGs. That sounds like a recipe for unchecked inflammation.
It certainly points towards an imbalanced, overly inflammatory state in at least some dogs with CIE.
You also mentioned STAT3 activation earlier. Can you remind us what STAT3 is and why it's relevant here in the adaptive response?
Right, STAT3, it's a really key molecule inside cells. It's a transcription factor, meaning it controls which genes get turned on or off. And it's involved in lots of processes, cell growth, survival, but importantly, also inflammation, often downstream of cytokines like IL-17.
Okay, a critical switch.
Yes. And finding that STAT3 is upregulated, meaning it's more active in the duodenal lining of CIE dogs, particularly those with the more severe immunosuppressant responsive form, IRES, is significant. It's been proposed as a potential clinical marker for active inflammation in the gut lining. Monitoring STAT3 activity might help us gauge how active the disease is.
Another potential marker then, moving beyond T helper cells, are there other adaptive immune cells playing a role in the gut itself?
Definitely. We need to talk about intestinal intraepithelial lymphocytes, or IELs.
IELs, where are they found?
As the name suggests, they live within the epithelial layer, the very lining of the intestine. They're right there in the front lines.
Like guards embedded in the fortress wall.
That's a perfect analogy. And a specific subtype of these IELs called T0 plus T cells have been found in increased numbers in dogs with CIE.
Gamma delta T cells, okay.
Yes, and interestingly, that's similar to what's seen in humans with Crohn's disease. It suggests a heightened level of immune surveillance, maybe even a misdirected response happening right at the gut surface.
Okay, what about the antibody producers, B lymphocytes, and the antibodies themselves?
They're definitely involved too. We see increased numbers of B lymphocytes, the cells that mature into antibody-producing plasma cells, both in the bloodstream and within the gut tissue of dogs with CIE.
And the antibodies they produce.
We see higher levels of plasma cells producing certain types of antibodies, specifically IgG plus EAD, IgG3 plus seed and IgG4 plus field. These are all indicators of an active, ongoing adaptive immune response trying to target something.
But hang on, I remember reading something slightly paradoxical about IgA antibodies. You'd expect all antibodies to go up with inflammation, but isn't IgA sometimes reduced in CIE?
That's a sharp observation, and yes, it highlights the complexity again. IgA is the main antibody type that protects mucosal surfaces like the gut lining. It acts like a first line of antibody defense right there in the mucus.
So you'd think you'd need more of it.
You would think so. But several studies have actually found reduced levels of IgA in the gut lining and even in the feces of dogs with CIE.
Why would that happen?
The thinking is there might be a defect in something called IgA class switching. That's the process where B cells switch from making other antibody types to making IgA. If that process is faulty, you might not produce enough IgA for adequate mucosal protection.
Okay, so potentially less protective IgA, but is it always low?
Well, to add another layer, some studies do find higher levels of specific IgA directed against certain bacterial components. So the immune system is trying to make IgA against some gut bacteria, but maybe the overall production or function is impaired, or maybe it's targeting the wrong things. It's not entirely clear-cut.
It's definitely complex. Now, you also mentioned IgG antibodies. I understand dogs with CIE have more IgG sticking to their gut bacteria, IgG fixation.
Yes, that's another key finding. Dogs with CIE tend to have significantly higher levels of IgG antibodies bound or fixed to the bacteria in their gut, especially bacteria belonging to the actinobacteria group.
So IgG is basically tagging these bacteria. What's the consequence of that?
You might think tagging helps clear them out, but it seems to backfire here. When other immune cells, like macrophages, encounter these IgG-coated bacteria, it triggers them to produce more pro-inflammatory signals, particularly TNF-in.
So the IgG tagging actually fuels the fire.
It appears to, yes. Instead of neutralizing or helping eliminate the bacteria quietly, the IgG coating seems to provoke a stronger inflammatory reaction from macrophages, contributing to that vicious cycle of inflammation in the gut.
Okay, so bringing this all together now, innate and adaptive systems. It's obviously not two separate things happening, right? They're constantly talking to each other.
Absolutely integrated.
Yeah.
They communicate nonstop through signaling molecules, cell-to-cell contact. It's a highly interconnected network.
So painting the big picture of CIE, how does this breakdown in communication, this crosstalk, lead to the chronic disease we see? What's the overall story?
Well, the current thinking goes something like this. It likely starts with that breakdown in the gut barrier integrity we talked about, maybe combined with a loss of that crucial immunological tolerance towards the normal gut microbiota.
The leaky wall and the overreactive guards.
Right. This allows bacterial components or even whole bacteria to cross the barrier, which triggers an immediate, exaggerated innate immune response.
The first responders rush in, maybe too aggressively.
Exactly. And that innate response then sends out signals, cytokines and other molecules that activate and shape the adaptive immune response. They call in the specialized forces the T cells and B cells.
Which then might also overreact or target the wrong things.
Precisely. The adaptive response gets skewed, maybe producing the wrong types of cytokines, maybe failing to regulate itself with enough dreg cells, maybe producing antibodies like IgG that actually worsen inflammation. It becomes a self-perpetuating cycle. The inflammation damages the barrier further, more bacteria get through, triggering more innate response, which fuels more adaptive response, and round it goes.
A truly vicious cycle. And you mentioned earlier the challenge in dogs is that pinpointing one single dominant pathway like Th1 or Th2 is much harder than in human IBD.
Yes, that heterogeneity is key. The specific balance of innate and adaptive responses seems to vary a lot between individual dogs, depending on their genetics, their specific gut microbes, the type of CIE they have, FRE, IRE, etc. That makes finding a one-size-fits-all explanation or treatment really difficult. Researchers are still very much piecing together all the different ways this cycle can play out in dogs.
It really highlights why it's such a complex condition to manage.
It absolutely does. It's not just one switch flipped, it's a whole cascade, a network effect.
So as we start to wrap up this deep dive, it's clear that, wow, while we've learned a massive amount, there are still pretty significant gaps in fully understanding CIE.
Definitely. Especially nailing down the precise roles of, say, barrier function versus loss of tolerance versus the specific microbial triggers and how they initiate the whole process.
And many of the exciting targets being explored for human IBD treatments, particularly related to the innate immune system, they still need a lot more investigation, specifically in dogs, right?
Exactly. We can't just assume what works in humans will work the same way in dogs, given the differences we've discussed. We need dedicated canine research.
But this kind of deep dive, exploring all this complex immune stuff, the immunopathogenesis, is not just academic, is it? It has real-world implications.
Absolutely, not just academic. Understanding these intricate pathways is fundamental if we want to find better tools. We need more reliable biomarkers for diagnosis and prognosis. We need specific molecular targets for developing new, more effective, and potentially safer therapies for these dogs.
Discoveries here are really the key to improving how we help these animals.
They hold the key, yes. Better diagnostics, more accurate predictions of how a dog will do, and hopefully targeted treatments that address the underlying immune dysfunction, not just the symptoms. That's the ultimate goal.
Okay, so to leave our listeners with something to chew on, considering everything we've talked about, the leaky barriers, the confused immune cells, the shifting bacteria, the genetic predispositions, the complex signaling, if you had to pick one thing, what do you think is the single most crucial piece of this puzzle we need to crack to really make a breakthrough against CIE in dogs?
Wow, that's a tough question. One single piece.
Yeah, if you had to choose the linchpin.
I think maybe understanding exactly what triggers the initial loss of tolerance to the gut microbiota in susceptible dogs. Is it a specific bacterial shift, a primary barrier defect, a genetic glitch in immune recognition? If we could pinpoint that initial trigger,
Then maybe we could intervene much earlier.
potentially yes. Or develop strategies to restore tolerance. And figuring that out in dogs might even shed light on similar complex inflammatory diseases in humans where tolerance is also lost. It's all interconnected.
Fascinating. Understanding that first domino, well, that's certainly something to